Hi, everyone. In the last chapter, we explored what microbes are and why they matter. But for most of human history, people had no idea microbes even existed. People saw food spoil, they saw diseases spread, they watched bread rise and wine ferment, but they could not see the organisms responsible. In this lecture, we're going to explore one of the most important technological breakthroughs in biology, the invention of the microscope, and how it revealed an entirely invisible world. By the end of this module, you should be able to explain how the invention of microscopy revealed the previously invisible world. Long before microbiology existed as a science, humans were already interacting with microbes every single day. Bread rose because of yeast, milk soured, wine fermented, diseases spread from person to person. People understood the effects, but not the cause. To ancient humans, these processes often seemed mysterious or supernatural because the organisms responsible were too small to see. At this point in history, microbes were influencing nearly every aspect of human life while remaining completely invisible. The problem is that the human eye has physical limits. Most humans can only resolve objects that are around 1/10 of a millimeter in size. That's zero point 0.1 millimeter or about 100 micrometers. That may sound small, but most bacteria are only about 1 to 5 micrometers long. That means bacteria are far below what the unaided human eye can detect. So even though microbes were everywhere, humans literally lacked the ability to observe them directly. To reveal the microbial world, humanity needed a completely new tool. In the late 1500s and early 1600s, early microscopes began appearing in Europe. These early compound microscopes used multiple lenses to magnify objects. For the first time, humans could enlarge tiny structures that were previously invisible. These microscopes were far from perfect. Images were often blurry, distorted, and difficult to interpret. Still, they represented a revolutionary idea. Technology could extend the limits of human vision, and that idea would permanently change science. One of the most important figures in microscopy was Antony von Leeuwenhoek. Unlike many early scientists, Leeuwenhoek did not rely on complex compound microscopes. Instead, he built incredibly high quality single lens microscopes by hand. These simple instruments produce remarkably clear images. Using samples from pond water, teeth scrapings, and other materials, Leeuwenhoek became the first person to directly observe living microorganisms. He called these tiny organisms animalcules, meaning little animals. For the first time in human history, someone had discovered an entirely hidden universe. The invention of microscopy transformed biology. Scientists could now directly observe cells and microorganisms instead of simply guessing about their existence. This shifted biology from speculation toward observation and experimentation. Eventually, microscopy allowed scientists like Pasteur and Koch to connect microbes to disease, fermentation, and many other biological processes. In many ways, modern microbiology became possible because microscopes allowed humans to finally see the living world at microscopic scales. Another major figure in microscopy was Robert Hooke. Using compound microscopes, Hooke examined thin slices of cork and published detailed drawings in a book called Micrographia in 1665. When observing cork, Hooke noticed tiny box-like compartments that reminded him of small rooms used by monks called cells. That observation introduced the term cell into biology. This may seem simple now, but at the time it was revolutionary. Microscopes were revealing structures and forms of life that humans had never imagined existed. As microscopes improved, scientists realized that making objects larger is not enough. Magnification simply makes an image appear bigger. Resolution refers to how clearly details can be distinguished. A blurry enlarged image is not very useful. Good microscopes require both high magnification and high resolution. Understanding that difference is essential for understanding how modern microscopes work. And that's what we can see here. In the left-hand group of images, we see a low magnification and a high magnification. And in the right-hand group of images, we see low resolution and high resolution. Notice how in the low resolution, it's the same size as the high resolution, but in the high resolution, you can distinguish clearly the different the different structures on the object. All right. So before microscopes, microbes were hidden from humanity. After microscopes, biology changed forever. In the next lecture, we'll explore how modern microscopes work and how scientists use light and lenses to visualize the microbial world in extraordinary detail. See you there.